course. The main idea behind the course is that we will have, we start from the beginning, we start fresh. This is a course that is supposed to be targeted towards the LSEC students, the graduate students, which means we start very basic and we go through all the, or several of the sensory systems. We have a talk or two which is basic sensory physiology, and then we try to ramp it up a little bit to more advanced. And you're supposed to give some talks in the middle. Now the problem is that this has become more than an LSEC course. It's the first course out of two. The second one is given by Mickey London. It's called Physiology B and has to do with motor system. And together it will give you some general framework of the sensory and motor system together. Now, are there any undergraduate students in this course? You are, Ralph? Okay. So you are the only one out of that is allowed to take the course because of special circumstances. And who is not from the LSEC program? Who is not from the LSEC program? All the rest are LSEC students. Wow, okay. So how many is that?
70.
70. Wow.
70?
Okay, so the three of you, which are not from the LSEC program, are not going to give talks. Instead, you're going to hand in a paper, a written paper. Well, including you. And what we'll do with the 17, I don't know. Two of the students, we're going to have, we have 5 sessions, seminar sessions. 2 of them are going to be 4 lectures. Okay? And you will have to set the schedule yourself, or I can do it, no problem. Each, and so what are the demands of the course? These frontal lectures, you do not have to attend if you don't want to. No problem. I'm going to start on time and try to finish on time. After 45 minutes, we'll take a 15-minute break, and then another 45 minutes, and hopefully an open discussion at the end. Recorded? What? Recorded. I think it is recorded. Do I need to put on the microphone? מישהו יכול להביא לי את המיקרופון מהמירותי? תודה. So the lectures are recorded and you don't have to read. Much of the stuff that we're going to talk about, again, is basic. And you can find it mainly in the Kandel, Schwartz, and Jeffel textbook, and you'll have the chapters that are relevant to what we're going to talk about. Besides the lectures which are you can attend or not attend, as we will, we're going to have a few events that you should already mark down. The first event is called No Class. That is going to be a class cancelled. It's on. It's because of the course in Shitot. You're going to have one session of working in the lab. So. Okay. So in April 26th, this is the schedule. I'm going to put it up on the. I'm new in Macintosh, so excuse me. Okay, so April 26th is going to be allocated to Shitot. It's a Sunday, and we're not going to have class. You're all going to be, or at least the students who take Shitot are going to be in our lab doing microscopy, whatever, hands-on stuff. Okay? That's one thing. The second event is going to be, discuss classic paper in 3 different topics. All of you must attend all the seminars by the students and these 3 sessions of discussing the paper, which on the first paper, the classic Hubel and Wieser, we're going to allocate 2 hours to that. The rest of the papers, we're going to allocate 1 hour. And I'm going to publish the names of the paper. You all must read the paper and be part of an active discussion, which means that you're going to tell us about the papers, and I'm not going to teach it, but rather you must read and follow and understand what's in the paper. And if you haven't, tell me about it, and I'll take 1 point off of your grade. The grade is as follows: 80% is an exam, final exam. 10% is your seminar or the paper that you're handing in. It's going to be graded from 1 to 10 on the talk that you're going to give, and another 10 points is the attendance of these seminars and papers. Okay? You can skip the whole course, but if you're not going to be here on the date that you must attend, we're going to take off points off your grade for that. Okay? So and you'll have all the dates here. There may be one small change. Okay, so there may be one small change. This is basically we're here today. Today and next time we're going to talk about just very, very general introduction. And if we will have enough time, next time I'm going to give you some tips about how to give a talk for your seminar, because this is something that people ask for. Again, you don't have to attend if you don't want to. And then we're going to start the different topics. The first topic is not really sensory systems, but basic neuroscience. So Idan Segev is going to tell you about single neurons, and he is having 3 sessions, and then we're going to have this classic paper discussion with me that you must attend. Idan then is going to give, there's a paper seminar here, and we're going to allocate who is giving what, which session in terms of the students. And like I said, you must all attend. We're going to have 1 session for Idan. Then Shaul Often is going to tell you about the visual system, and we're going to have another session. Mickey London about the somatosensory system, and I'm going to teach about the olfactory system and the auditory system. Okay? And basically that's it. That's the structure of the course. So you must be in the exam, 80%, give a talk or hand in a paper, another 10%, and attend the seminars and the paper discussions, another 10%, just attending and reading the paper. Okay? Question?
Hot.
The seminar is presenting a paper or
Yes.
giving a talk on a topic?
No. So the seminar is presenting a single paper. So each of the teachers will give will give 3 papers or 4, because we have 2 sessions. Because you're 17, so 3 or 4 papers, and these must be presented in 25 minutes in a regular seminar. Okay? Great. Any questions? Everything's crystal clear. Okay? So we can start. So this is introduction. You can find what I'm going to talk about in chapter 21 of Kandel and Schwartz. And we're going to talk about sensations and sensory systems. So what is sensing? For example, vision. Seeing is. What is seeing? What do I mean when I say seeing?
Getting light signal as an input from
Okay, so that's not enough. So a light must hit your retina. Okay, fair enough. Okay, so having light fall on your retina and somehow perceiving the light that fell onto your retina. Okay, any other ideas? Everybody's happy with this, yeah. Mm-hmm. So if I ask you to close your eyes and you imagine some image that's uh, consistent with the world around you. Okay, so we should see for something for behavior. Okay. I see. Okay, so cleaning up what's coming in order to make sense out of it. Fair enough. We are going to see everything that people talked about or raised up here along the way. But we're going to start with David Marr, which was a famous psychologist and philosopher, died at the age of 35, that is saying that seeing is knowing what is where by looking. That is the definition of David Marr, and we're going to change this definition or look at this definition as we go along. David Marr described that in order to understand a sensory system, we must understand it at 3 levels of analysis. The first one is to understand the problem. What is the computation at the computational level? What is the problem that the system has to solve? The second level is an algorithmic level. How does the system solve the problem? Okay, so first we have to define what is the problem, and then we must understand or try to understand the algorithm that is used to solve this. And at the last level is how does the system actually do it? Okay, because we can ask a camera to send some picture, and we know what the problem is. We can define it computationally very easily, and we can sit down and write algorithms to solve this problem. This, of course, does not mean that the brain is doing it this way, although many computational neuroscientists like to think so. They're sitting very solidly in the algorithmic aspect, but whether the brain actually does it in the specific algorithm that we think that the problem can be solved is a whole different story. But by David Marr, if we are able to solve what is the problem that should be solved, what is the algorithm that is used to solve it, and how does the system actually do it, we will be able to fully understand a sense. And again, as we go along, we will see what kind of tools and what kind of knowledge we have in the different senses. So what is the problem that we must solve in vision, for example? By David Marr, the problem is to transform a 2-dimensional image that falls on our retina into a 3-dimensional reconstruction of the visual scene. This is by David Marr, and now we have a more modern version of this older definition of what is the problem that we have to solve. So this is courtesy of Peter Latham, which Eli Nelken used to teach this course. So Eli took it from Peter, I took it from Eli. So all thank yous are done now. And the problem, or the more modern version of the solving the problem, is not to reconstruct a 3-dimensional scene from an image, but rather to recover the latent variables in the image. So when we say seeing, is that this thing is falls on your retina, and now you have to recover what is in this image. What is in this image? So some people like you will say there's a house, there's a tree and a sun. Some people will say a blue color. Some different people will say different things. But the idea is that we can, we should recover the latent variables from the image. Okay, and that's the idea or the problem that we have to solve. And the nervous systems actually solve it. And of course, it can be a completely different image that falls on your retina, which gives you the same information. Okay, so for example, in the next image we have, if you look at these images, you will say this is Sarah Netanyahu. Okay, but of course the pixels that fall on your retina are completely different in each and every image. Okay, you can have the same face looking at different angles, but still you will recover the same aspect from this image. Or you can have, you know, happy, sad, serious, whatever, different lights. In terms of what falls on your retina, it's a completely different story. But we have to recover the latent variables from that. Okay. So so the problem, the modern version of sensing is reconstructing the latent variables. And there are many, many algorithms that you can solve. Again, if you're part of the computational community in Hebrew University, you can think of different models in vision. There are many, for example, graphical models that are common. And again, the idea is to pick up the latent variables from this low-level representation that falls on the retina. And from these low levels of representation, the algorithms must infer the latent variables. We must infer, and this inference can be shaped by different aspects of the physiology of the animal. Okay, or what it is one has to infer from these images. What do you mean by latent variables? The latent variables is that there is something in the image, okay, and you're not. The idea now is not to put this triangle together with the square, or put together, reconstruct a sun from different variables, but rather to pick up what is hidden, the latent, the quiet variables in the image. Okay, that's the idea of recovering these variables from the image. Okay, sorry. And in terms of implementation in neural networks, like I said, we can be very smart on defining the question, which is very important. An algorithm, we can have many algorithms, and whether and how this is implemented in neural networks, we still have no clue. So, welcome to neuroscience, like people have probably told you. We have lots of work to do because we still have no clue how the nervous system can extract the latent variables from an image. How do we know it's Sarah Netanyahu there? Although computer vision can do many, many things. It can take us very far. It still does not mean that the brain is implementing this task in the same way. Yes? No, no, no. It's generally true. So our best knowledge is in vision, by far, and we will see. I mean, I'm talking about the full task. We have pretty good idea what's happening in the retina and questions about resolution, etc. We'll talk about that. But to answer, for us to fully understand a system means that we know how the two-dimensional image on the retina is fully interpreted, or the bouquet of odors is completely understood as this smell or that smell, etc., etc., and other senses. Okay, and we still don't know that, and many people are still working on sensory systems. And if you compare how we understand heart, for example, we are still years away, even in the visual system, which is the most understood system by far. Okay, so we still have no clue, and this means that nobody will fire you, and you still have vision to be hired as a professor in the university and solve problems in sensory systems. Yes?
Why did they change from the David Moore's talk to the
it's a more correct version. We'll see now other people who think that, like your name? Tomer? Carmel said. And we'll see this thing changing. Well, it's a more, because it's not about reconstructing the pieces into this kind of a puzzle, but rather to understand. I mean, I don't have a good answer for you. This is the modern way of looking at the problem. Okay, yeah.
I would argue that reconstruction of latent variables, I mean, it's important in terms of, like, obviously it's something that you have to do as part of cognitive processes, but I'm not sure that it's necessarily also part of the definition of seeing it, because you can see a picture of something that doesn't really have any real latent variables. Like, you can look at a Jackson Pollock painting or Gaussian white noise or something like that, and you can still see, even though there aren't objects there that are latent. So I think that the latent variable reconstruction is important, but it's not necessary for what we consider vision.
Okay, so again, we will, as we go along, we will see how this develops. Some people say that the full definition must include what Carmel said, is we see in order to do something, that the sensory action loop must be complete. But you say if I open my eyes and something falls on the retina, I'm still seeing, and that still counts. A baby that opens his eyes and knows nothing about the world, so to speak, is still seeing something. He doesn't even have to recover anything. As far as as far as you are saying, it can be neuronal activity in the visual cortex. Yeah, maybe? That's for you seeing?
Well, I mean, there also has to be perception, whatever that means, but it doesn't.
Okay, so it has to involve perception.
Yeah, but it doesn't have to be in the sense of recovering latent variables. So I think that's a higher order cognitive process that doesn't exist in every situation of seeing. Sometimes there just aren't.
But you can't separate perception from attention, for example, because you don't know where the line is.
Well, I don't know if attention is anything to it. I'm just
Because
saying that
attention is like inhibiting the non-important data or whatever, as far as seeing.
If you are looking at that like whatever your thoughts about that, how this is
no, but my thoughts are a reflection, though. In other words, what I see is what I see, and then maybe I'll think about
But
it.
you
Maybe I'll
don't
think
see
about
all
something
things. You
else.
don't see all things.
No, no, this is good. This is good. Your name?
Tovia?
Tuvia?
Yeah.
Achla. Tovia.
Like the Tova.
Tovia. Okay. No, this is good. This is good. I want to make you angry and nervous. Huh? That's the whole idea of the thing. There's no complete true or untrue. I'm just saying how currently we look at the scene and where perception stops and cognitive processes kick in. For example, just imagining an image in your brain, I think there is a general argument that this is a cognitive process rather than a sensory process, the way we are thinking about it here. Okay, but what we are going to talk about is from sensation to perception, including perception. And if you say my thought, I don't even perceive what I see, I don't know. I'm a little unfocused about your definition, just a bit that you're unhappy with this definition, which is fair. Yeah.
I think maybe what you mean is kind of like when we hear, there's like the difference between hearing and listening. You say, oh, I hear you, but I'm not listening.
Mm-hmm.
So you receive noise and you apprehend that there's some noise, but you don't make sense of it.
Mm-hmm.
So maybe that's what Toby is saying, that you can receive light, but you're not making.
Well, this is fine to some extent, as background noise or something. But when we say in sensory systems that we want to understand the system, we want to understand the system that will lead from the physical variables to perception. Okay? That's what we want to understand as neuroscientists. How do we perceive something? Or how can we, in this case, how do we extract some latent variables from the sensory scene? I agree that there, if you hit with light on your, with pressure waves on your cochlea, there will be some activity in the brain. And when we are asleep, do we sense things or don't we sense? It's a wonderful discussion. But let's start from the basics, okay? Okay, so when we talk about sensory systems, we talk about sensation, which is getting the information into our brains, sensing it from the environment. We will talk about physical measurement devices, the devices that the sensors are using in order to detect these physical variables. These are normally in the sensory periphery in classical neuroscience. These are not the only sensors in the periphery that we have, and we will mention them as we go along a little bit. I mean, we talked about things like that in physiology, in basic physiology course, the ones of you who took it. There are many limitations in sensory systems which we have to consider. Physical limitations, the physical limits of the physics of light and sound, we'll talk about that. There's statistical constraints that we will talk about. And there are, of course, neural constraints, which is something that Omri talked about. The filtering of the system, okay? Our neurons are filters in space and time, and we'll talk a little bit about that. And perception, to perceive something, is to, again, extract the latent variables, although many people are not happy with the word, with this definition of perception, which is fine. Okay, so again, when we talk in neuroscience about sensory systems, it's from the sensation to the perception. Okay, that's what we want to extract. This is a nice image that I like in olfaction. You know, what is she perceiving here when she smells the flower, in this case? Okay, so let's start. When we talk about sensory systems, we think that we have direct access to the physics of the outside world. We think we hear everything, we see everything, but in fact, we have, or we must realize, that we have a very limited access and a highly filtered, interpreted version of the real world out there. If we go back to this slide, today for a few $100s you can buy easily detectors, or a few dollars detectors that can see far better than we can see in terms of the physical spectrum. We, for example, cannot see in the UV or IR. This is true for sounds as well. You will see that our ears and auditory system can have access to a very limited part of the world. Also with chemical sensors, this is a gas chromatography machine. With gas chromatography, you can see and detect very few molecules in the air, which we cannot do as well with our nose. Okay, so we are actually exposed to only a very limited, filtered, highly interpreted version of the world. And when we talk about perception, when perception is obviously in contradiction with what we think is reality, then we start to see things that look like illusions. Okay, and I'll give you some examples. Sound po. This is the sound. Hello, the HDMI. Okay, whatever. Maybe the Macintosh is strong enough. Okay. The last thing before I show you some illusions is what Carmel said, that also very common now is to close the loop of the sensory action loop, and people are talking about perceiving for something. We have active perception, and perception and action are tightly linked, and this link is highly important. So if we started by seeing is knowing what is where by looking, by David Marr, this is again a more modern version where people who like to describe basic concepts, Reza Shadmer, for example. Seeing is predicting what is where, verified by looking, in order to do something, okay, in order to close the loop. Again, this place at the Hebrew University is very strong in this kind of sensory action loop, has been working on these problems for many years, and one of the leaders in this field, both theoretically and to some extent also experimentally. Okay, So what we want to know is how is sensation transformed to perception. We will look at the peripheral encoding of the physical variables. We know quite a bit about how the very low-level sense organs process the information. We will look at hierarchy of brain areas, how this information is transformed as we go along. And you will see that, first of all, we lose information as we go deep into the brain. We split information as we go into the brain. And the information as we go higher in the sensory processing is becoming abstract as we go deeper. Okay? And like I said, when we have contradictions, let's say we usually have conscious access only to higher levels of processing. We'll talk about that. So when you looked at the image, you saw a house, a sun, and whatever, and not, small pieces of lines. And at that level of perception, things become rather abstract. And again, this can these conflicts can lead to strange effects. So for example, just to give you an example, there's a sign now, and you have to read what is on the sign. Ready? Okay. What was written there? One by one. So I'll give you again the same thing. Okay. Okay. Ready? Okay. So you know. Yeah, I see. So you know that. Okay. So let's take this out. No. What's this? Okay, I'll just fix this 1 second. Okay, so those of you who are sharp saw or could see that this road is slippery when wet, but when people normally read it fast, you see that they read or in many cases they miss the is and when, and see it as one general percept. The road is slippery when wet. Okay, now let's look at the colors. You have to name the colors that you see out loud. You did this already. Okay. Okay, and some things are dramatic. For example, this. So we must have the... Can you hear?
But look what happens when we change the picture. And yet, the sound hasn't changed. In every clip, you are only ever hearing ba with a B. It's an illusion known as the McGurk effect. Take another look. Concentrate first on the right of the screen. Now to the left of the screen. The illusion occurs because what you are seeing clashes with what you are hearing.
In the illusion, what we see overrides what we hear. So the mouth movements we see as we look at a face can actually influence what we believe we're hearing. If we close our eyes, we actually hear the sound as it is. If we open our eyes, we actually see how the mouth movements can influence what we're hearing.
It's a bizarre effect, Remember, the only sound you're hearing is bar with a B.
Ba, 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 ba.
What's remarkable about this illusion is even knowing how it's done doesn't seem to make a difference.
Ba. The effect works no matter how much you know about the effect. I've been studying the McGurk effect for 25 years now, and I've been the face in the stimuli. I've seen stimuli thousands of times, but the effect still works on me. I can't help it. The speech brain just takes in that information and doesn't care about what outside knowledge you bring to bear. Ba, 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 ba.
The McGurk effect shows us that what we hear may not always be the truth.
Ba,
But it also helps us to understand what happens when our senses conflict.
ba, ba, ba. When the brain has the conflicting information, it tries to make sense of that conflict. And depending on what type of modality is providing more, salient information, that information might override or at least combine with the other information.
So we can't always trust what we hear, because sometimes our sense of vision takes over, enabling us to maintain a coherent view of the world.
Okay. So the McGurk effect, very powerful effect. And again, just tells you that what you hear, what you see. Oh, sorry.
Hey, Vsauce, Michael here.
What's it?
Appears blue. This
Okay. Bon. So what we see or what we perceive may be different than what falls on our retina and what falls on our ears. And there is much more to the perception than just simple physical variables that fall on the retina, and it involves deeper processing of circuits in the brain, which are then interpreted or inferred by us, by the brain in some way. We will deal in chapter 22, mostly chapter 21, which is the introduction, what we will do now and later on, mostly with low-level aspects of sensory coding and looking at the standard model of sensory systems. Different people can think, you can see different definitions, again, more advanced. We're going to start with the basics. Okay, so how many senses we have classically? Five senses. This is, we all know, we call them the somatosensory system. These are the bodily sensations: vision, audition, smell, and taste. Okay, what makes a sense? Why do we call vision vision or audition? Why do we split it into different senses? Because the physical variables that are used to detect it, mostly true, and the sensors that were used to detect it, etc. Okay, so we all know that vision is not audition is not olfaction, and the physical variable that is measured is critically important. Sensory information is the neural activity originating for stimulation of the receptor cells of that specific part of the body. And again, you will get to know the different part of the body and the different senses. Okay. The physical measures, variables that we do measures come in different forms. They are normally different types of energies. It can be electromagnetic energy. For example, electromagnetic waves. In vision, we see electromagnetic waves. Some animals can sense different things, magnetic fields, electrical fields. It doesn't mean that humans can sense all of these energies. We can sense other types of energies like mechanical energy, things like differentially on one point of the body, so depending on which sensors is being where. Things like wind, water flow, pressure waves, like in sounds, it's pressure waves. We can sense things like heat and other things which do not count as the 5 classical senses, like sensing gravitational energy and inertia, and chemicals as well. And we understand different sensory systems to different levels. Okay? So there are other, within the somatosensory system, we talk about modalities, and which are not different senses. Okay? For example, pain. Is pain somatosensation or isn't it somatosensation? Or proprioception. Is it classically like, feeling touch? So these are modalities, are different modalities that we think, or thermosensation. If something is hot or something is cold. These use different sensors, different receptors in the body, and we sometimes think about modalities within the somatosensory system. So the somatosensory system is composed of not only, you will learn about the classical somatosensory system, the receptors on our skin, but there are other submodalities within the somatosensation. We will hear one talk during this course about pain. Okay, so the five senses, somatosensation has different modalities. It can detect mechanical changes, heat, chemical changes, okay? And vision is electromagnetic radiations within a very limited range in humans. Different animals see different ranges. In the auditory system, we detect pressure waves. In smell and taste, it's chemicals. Okay? There are other systems that we do not have, but many animals do. Sorry. First of all, there are more senses than the five classical senses. For example, the vestibular system, is it a sense or not a sense? So we do sense gravitation and inertia. The vestibular system sits in our close to our auditory system in the same region of the cochlea. We will not talk about the vestibular system, although it's actually one of the systems that we do understand quite well. And there are submodalities. Okay, there are other senses that humans do have. We can feel vibration. Like I said, proprioception, which is not the classical touch, but rather knowing the spatial relationship of the different parts of the body. We can sense temperature. We can sense pain. These are part of the submodalities of somatosensation. And there are also other senses. Okay, some of them are conscious and some of them are unconscious. For example, in physiology, we talked about thirst, I think, and there are sensors in our body, like mechanical sensors of blood pressures and osmotic receptors in the body that can sense changes in the volume. Many of these are not conscious senses, but nevertheless, they are responding to some cue in the environment of the body and operate on some mechanism within the brain. Okay, so these are also senses. You have to tell me if I'm at 45 minutes. I am. Okay, so let's just wrap this up. You'll get all your time back. Don't worry. Okay, senses that we do not have. We cannot see beyond the physical variables that limit our sensory system. We cannot sense other variables, and we cannot use the same variables differently. Okay, we cannot see with our ears, etc., etc. And there are senses that we do not have, and other animals do have. Echolocation. What? I'm a 45 on the sharp. Soon we'll take a break. Senses that we don't have, echolocation, for example, which is detecting objects and distances by sound. You can, bats, for example, or other animals work with sonar, biosonar. They emit sound and use the echoes to locate themselves. Vibrasa systems, those of you who are working on animals know that they have a very developed vibrasa system. We cannot detect what? Vibrasa system is the mustache, the whiskers, to use as a somatosensory system. Okay, so this is something we don't have, but other animals use. pheromones is something which is very potent in other animals and is not developed in humans. We'll talk a little bit about that. Like I said, seeing beyond below UV and beyond IR, electrical fields, magnetic fields. I don't know, some turtles use magnetic fields to swim in the ocean, and we don't use the magnetic fields, etc. One thing you have to remember or to keep in mind is that if we look far enough deep into each sense is species specific. Okay? We cannot say that vision in any species is exactly the same, and there are many variations on the same theme. Okay? So this is like a general introduction to the introduction, and let's take a break and meet here at 1:02. כן, בהחלט. לפעמים אני מוסיף ומשנה דברים, אז בדרך כלל אני עושה את זה או בבוקר שלפני ההרצאה או בערב שלפני, אבל כן. לא, לא, אין בעיה. אני אגיד לך מה, הבעיה שלי מה? כן, 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 כן. כן, לא, אני פשוט... הקורס הזה היה משפחתי של 12 תלמידים, וכל תלמיד עשה 3 סמינרים. זה היה טוב לחבר'ה של החישוביות שבאו ממקומות אחרים. זה היה הרעיון. הרעיון הוא ליישר איתם קו. ואז הוא גדל וגדל, ופתאום היו 50 אנשים בכיתה, ואי אפשר היה לעשות יותר... קיצור, הגבלנו אותו רק ל-graduate students.
מבחינת המספר של התלמידים.
כן, 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 כן. אז בסדר גמור. אין בעיה.
phone doesn't stop ringing. Somebody's looking for me. So I turned it off. So I don't know what time it is. Hello, wake up. So? עליי? שבע. לא? עליי? Okay. Great. So, sensory physiology, the modern sensory physiology, the way we see it today, starts at the 17th and 19th centuries with the empiricist and the idealist philosophers that argued about how knowledge is represented in the brain. The empiricists, which were represented by Locke and the theory of mind, argued that the mind is a blank slate and knowledge is obtained only through experience. So when a baby is born, it's basically empty in there in terms of sensory perception or how the sensory world is represented in the brain, and knowledge is with time obtained through the experience of the brain. On the other side, Kant, which was the idealist, argued that the brain has a priori knowledge which was evolved in the structure of the neuron of the brain itself. They didn't know about neurons at the time. Thus, it's not only the stimulus but also the brain inherent properties that contributes to the perception. A little bit later, this was more commonly known as the nature versus the nurture argument. This is, we know today, is somewhat a redundant argument because one knows that if you take a newborn animal and close it without any senses, any sensory experience, it will not evolve and develop to how the brain is now in an adult animal handles a sensory perception. So nurture is important. We also know that there is some limitations, very basic limitations in the properties of neurons and circuits that determine how we perceive sensory information. But this is again an historical note. And those initial thoughts later on evolved gave rise to the topic of what is known as psychophysics, even today, which is the relationship between stimulus and the sensory experience. And to this date, this is still alive and kicking, psychophysics. And later on, only later, in our times, 1950s and higher, where people have discovered the ability to record from neurons. We'll talk a little bit about that. A new field has developed, which is called sensory physiology, which is basically the relationship between the stimulus and how it is processed by the brain. And the good understanding, or a better understanding of the processes, comes when we can merge between these two fields, psychophysics and sensory physiology. And as you go along into more and more modern neuroscience, you see that these things are going hand in hand. Okay, the psychophysics and the sensory physiology. You had a question? No? Oh. So only in rare cases you see beautiful examples where psychophysics and physiology interact. And if you don't know this work by Yitzhak Fried from Tel Aviv and UCLA, they record from human patients with epilepsy. And they're basically searching for the epileptic region in the brain. And so they stick electrodes. They think where the epileptic focus is. They stick electrodes.
and they wait for the epileptic seizure to pop up. And meanwhile, they're with electrodes sitting in interesting places in the brain, so they do experiments. They show the patients movies, they play games, things like that. And this is a very beautiful example of neurons recorded. So for those of you who don't know and are not familiar, this would be a PSTH, post-stimulus time histogram, and these are the raster plots. We'll talk about the definitions soon. And in this case, these neurons were that they recorded in this human were highly specific. So this is shown very nicely in this movie. For those of you who haven't seen this, so this is a patient. Okay, so maybe I should explain. What you see here is time over the course of the recording, and this is basically the PSTH, the ongoing firing rate of the neuron. And this is the videos that they show this person. Okay? So we started by showing, post-stimulus histogram, time histogram. Okay? It's just a collapse average of the firing rate. We'll talk about that soon. Okay? But again, so we're recording from humans while he shows this person the videos, and later on he's asking him a question. Okay, so this firing of single neurons in entorhinal cortex while watching short videos. These are the videos. Okay, Hollywood sign, Martin Luther King, the Martin Luther famous speech, Madonna, Titanic film, yes, voila. Okay, from The Simpsons. Great, so he's just showing him the video. Okay, again, Martin Luther King. Titanic. So, this is pretty strong, huh? Every time he shows him the Simpson movie, this cell in the entorhinal cortex gets crazy. And this goes on and on and on. Okay, and now it's the same neuron, but now he's asking him. He's recollecting what he just saw. What are the videos that he just saw? And Hollywood sign that you showed me. Okay, so this is a recollection. He's not showing him any more movies. Okay, so this is entorhinal cortex. When he sees The Simpsons, in different contexts, this neuron is firing like crazy. But and also when he's recollecting about what were the images that were presented to him, also these cells fire like crazy. Okay? Now, does this mean we understood vision? Or understood, what have we understood from this? There's a big argument about the hierarchy of sensory processing. This is clearly on the top of the hierarchy if you talk about Simpsons. Okay? So this is clearly a Simpsons neuron. Even when he's just recollecting what he saw. I don't even know if he has an image in his mind of The Simpsons, but this is clearly a dramatic example of what is also known as grandmother cells. Okay? There are cells at the top of the hierarchy When they fire, you know it's Simpsons. Yeah. Yeah, so people looked at it. There's Jennifer Aniston cells and Michael Jordan cells and lion cells and face cells, etc. But you can find, so face, for example, there's a specific region in the brain that is recognizing faces, and neurons will fire, like I showed you, Sarah Netanyahu will fire to the face of Sarah Netanyahu. Some of these cells will also respond just to the name. So the very famous example of the Jennifer Aniston cells, even when you just see the text, Jennifer Aniston, these cells also start firing like crazy. But the question, have we understood vision with these experiments? Not yet. Okay? Okay, normally we have a very limited access to the stimulus itself, the sensory experience or the sensory physiology. Like you mentioned, maybe it's about happiness, attention, I don't know. So we don't have access as experimentalists to all the parameters. And you need to think, and we still argue about this to date, what does it mean to fully understand the system? I don't know. Idan Segev would say that if you can build a mathematical model of the system, then you fully understood it. Then we can put the keys and go back home. That's it. We understood this part of the brain or this part of the sensory system. Maybe you agree, maybe you don't agree, I don't know. But the question is what it is to fully understand. Now, to see the Simpson cells, did they fully, there are many stations between the retina and the Simpson cell. And if we want to fully understand it, do we have to understand the whole pathway, all the way through, all the cells, all the molecules that are involved, or maybe not? And you have to think about now that you're at the stage where you have to decide, maybe some of you already decided, what is the avenue that you want to pursue in science, in your thesis and PhD. How meaningful what you're doing, what the thing that you are doing is, how far it can take you in understanding a full circuit, okay? Do you want to work with Yitzhak Fried, put or record from electrodes? Do you want to look at mice when you record? Do you want to do modeling, compute something, whatever? Okay, so our goal is to fully understand the system. In this case, it's the sensory system. And what it means is not completely clear. And what my role is to bring you back to reality and not to only, it's good to dream and to think about what you can do and how what we can do in order to make real progress. But let's see what has been done until now in different sensory systems, and that's basically the goal of this course. So the first attempt was to understand simple stimuli and simple sensations and to quantify that. And this was done mainly in the level of psychophysics. Okay, before we could record from neurons and maybe understand it a little bit more physiologically, the beginning was quantifying sensory experiences. It started in 1834 with Weber and the famous Weber law. And Weber claimed that the just noticeable difference between two stimuli is proportional to the magnitude of the stimulus itself. So we can sense, let's say, whether we can sense 1 kilogram of metal. So if I ask you to differentiate between 1 kilogram and 2 kilogram, you can easily do it. But if I ask you to differentiate between 100 kilograms and 101 kilogram, you will not be able to do that. Okay? Our sensation or the just noticeable difference that our senses can sense are proportional linearly to the magnitude of the stimulus. This is Weber. Many still look at the Weber law today. You can find it in many textbooks, but this has some kind of evolution. So it evolved later. Every one of these laws lives between 20 to 100 years or something until somebody changes a little bit. Fechner's extensions of the Weber law says that it's not a linear function, but rather the intensity of the sensation that we experience, that the sensory system experiences, is some kind of logarithmic scale with some kind of constant, and log of S divided by S0, which is the threshold stimulus. And it turned out later, after many arguments, that this is an oversimplification. And there is now Stevens power law, which came up 80 years later. Again, look at how simple things takes them 80 years to evolve. All the rest of us are busy doing our daily regular experiments. So Stevens' power law proposed a relationship between the magnitude of a physical stimulus and its perceived intensity or strength based on some power law. And depending on the system, you can punch in the power law, and it can behave in different ways. And this also turns out today, if you ask a psychophysicist, this is also an oversimplification. But anyway, one thing that you see also today in psychophysics is what we call the psychometric curve. Those of you who don't know it, it takes normally this kind of form. On the x-axis is the intensity of the stimulus, and on the y-axis is the perceived intensity. It's a very common and highly used way to determine things like thresholds. And detection thresholds, for example, are normally determined by a 50% chance to determine a given stimulus. So here would, if I give you, let's say, a sound with a very low intensity, let's say, I don't know, 5 dB or whatever, dBSPL, you will not be able to hear it. Then I throw all the time and I tell you, can you hear it or can you not hear it? And that, at a given level, let's say intensity 10, you can perceive it 100% of the times. And one can always construct these psychometric curves, which will take this kind of structure, and we determine or we define the threshold of a stimulus by the 50% detection level. There are other ways of determining thresholds. There's a method called the method of detection of limits, or matching to a reference stimulus. You can take a stimulus and say whether it's the same or different, and one can start to calculate J and Ds. Another way or the involvement of quantifying perceptions, there are different ways if you look at the literature. The book says, gives us 4 different ways. One is nominal, so to give names without given ranks. This is common, for example, in the olfactory system, or historically, I would say common in the olfactory system, where people would categorize. How do you quantify an odor perception? Okay, so you can say if it's a given odor is strong or is it weak, but the classical way to quantify these perceptions was either to give them some kind of a category. So this were the classical 9 categories of smells. Okay, it could be ethereal, aromatic, fragrant, embryos. Some are very stinky, fruity, whatever, blah blah blah. Again, 100 years later, the new way to quantify perception was the minsara. How do you say minsara? The prism of Hans Henning, where an odor can be spicy or resonant or burnt on one axis, and you can place your perception somewhere on this prism. Today there are more modern, a little bit more modern way to put this, but again, categorizing perceptions nominally is one way. Ordinally is also something very very common, but it's highly subjective. So if you go to the doctor, many times you go to the doctor and he would tell you how much. You say you feel painful, and he would ask you how much from 1 to 10 do you feel painful. That's another way to quantify your perception. And you will say 7 or 9. This is very subjective. Also, it doesn't mean that the distance between 1 and 2 is exactly like the distance between 8 and 9. But so this would be an ordinal way of ranking. Highly subjective, but still something that is commonly used. Another way is counting, and this now gives a very clear quantity to the percept. Okay, how many clicks did you hear? So it's very clear that if you heard 3, if you heard 4, it's exactly 1 higher than 3, and 5 is exactly 1 higher than 4, etc. Okay, so this is a little bit more quantitative. And we can also do the ratiometric way of quantifying perceptions, which is some kind of combinations of the above, which you can normalize it to the subject. So one example is how much do you feel the pain, okay? So if we ask what is the intensity of the doctor asks you how much do you feel this, and he continues to push harder and harder and harder, you will say from 1 to 10, it's again 1, 2, 3, 4, 5, 10. And if it's normalized, it can have a more quantitative measure to quantify perception. And again, if you will join a sensory lab, audition, vision, etc., you will see that in the field this is still something not always written in stone and very clear, but different people use different methods to quantify perception. A nice example, which I specifically will talk about in the olfactory system, is what we just mentioned, these prisms and categories. So for example, Noam Sobel and Weitzman has a fancy way to categorize perception based on some principal component analysis. And each odor will have a specific place on this two-dimensional PC diagram. And you can say that one odor is at an exactly given distance from another odor based on this PC space. Of course, this is highly, people still don't accept it completely, but he's pushing towards better quantification of odors around us. Yeah, questions? No. Okay. Now, these psychometric curves can be affected. We saw what we call the ideal observer. We'll talk about that soon in this yellow curve. But these perceptions can now are affected by the state of the animal. So for example, if you look at these psychometric curves of a given animal, the same animal can have the yellow psychometric curve, but also this curve can be shifted in its, not in the structure, but to the left or to the right, if the animal has some bias to its behavior. So for example, if you know that you get, I don't know, money for each time you say that you heard the stimulus, and that there's no punishment, so your incentive is to have, you can just push, you don't care, okay, that you heard the stimulus. So you'll see shifts in thresholds. More people will say that they heard the stimulus, for example, or the other way around. If you know that the punishment is very severe for an answer that you give as a subject, then this thing can be shifted to the right. So this will change the number of false positives and false negatives that one can see when you quantify the perception. Normally in psychometric curves, for example, in thresholds, you will see these kinds of curves and behavior. One very good way to do it is to put in catch trials also. And you can determine easily the structure. And normally it will take up this form. The slope of this form can be very different. Also, it will be biased by the behavior of the... but psychometric curves are very common. Okay? Okay, and of course, most of our understanding, or our good understanding of the neuroscience, of the sensory systems, comes from animal work, where we can actually go into the brain, put our electrodes in, measure the synapses, measure the connectivity between the neurons, and really understand mechanistically what is happening in the brain. I mean, with all the respect to psychophysics and psychology, it can take us only so much on our way to understand. We must combine the psychophysics and the sensory physiology to get a better understanding of the biology of the system. So most of our understanding of sensory systems, or much, not most, but much of our understanding comes from work with animals. Okay, so how do you quantify perception in animals? That is not an easy task, because in many cases you cannot ask them what they feel or what they saw or imagine something, etc., which are the type of things we were talking up to now. But still, you can predict sensations by quantifying the percept using probabilistic statistics. This is a very common way to predict the behavior of animals. This is normally done in not only, again, but in many cases is done by assays, bioassays, where you have either a two alternative false choice task, which is known as a 2AFC, or a yes, no, or go, no-go task, where the animal has two answers to give. This probabilistic statistics was actually invented by psychophysics and used in the Army, in the American Army, to detect radar, to detect airplanes coming into the danger zone. And then you have to say, you have to give a yes/no answer. Is there an airplane? The radar has to give is there a radar? Is there an airplane, a Japanese airplane coming in, or is there not? And of course, you can see the importance of making a mistake here. Okay, so where you put your threshold exactly can be very critical. And this was, so the whole statistics of quantifying perceptions in animals that we use today is based on these probabilistic statistics, and I will elaborate on that a little bit now. Yeah. So it depends what you want to study, but generally speaking, the community is trying to minimize the noise and the variance in the experimental system that they are studying, generally speaking. The work in mice, for example, is done in genetically identical animals in many cases, and the reason is to reduce the variance and the noise in the system. In all biological systems, you will see variance. Sometimes this variance is important, especially neuroscientists like to inject noise into the spiking activity and things of that sort. But, you know, the real thing, depending what you're asking. If you're asking how am I perceiving the weight of the life experience, is not something you can measure by taking like no, you can measure. Why can't you measure? So now you are reducing the variables to the very basic phenomena that you want to study. Let's say maybe the phenomena is highly dependent on the life experience. So that's a different question. If you're interested to understand how does the experience change the way the sensory system is working, then there's advantages and disadvantages of working with a model organism which is genetically completely alike. Then the senses is like hardly working at all. Exactly. Okay. So now you want to know, so you're talking about a different question or a different aspect. You say each and every animal has different experience, which is what makes you different from me and us different from each other. Great. I need experience to see the, Yeah, but these animals, they grow in a cage and they have some experience, which is fine. Now, some animals will have different experiences, and then maybe you want to understand what is this particular experience did to the animal or to the sensory system, which is fine. But now, if I want to understand how the neurons in the cochlea or in the retina encode information, maybe at this stage, first of all, there's no change in the experience, maybe. I still wouldn't call it sensory perception, but you just need an input and output function, okay, of the cell. Now let's look at the next station, and next station, and next station. And what we know from animals is that there are many parameters that are very, very similar between each and every animal, regardless of the experience that you undergo. Okay? Regardless. For example, you just saw the McGurk effect. Each and every subject in this room had the same illusion or problem, okay? That you hear exactly the same sound, but you perceive it differently. It means that something is exactly the same for all of us. If I want to understand the McGurk effect, I can study humans, no problem. Okay? So, and there are advantages and disadvantages to work with exactly the same species. Okay, so in this kind of, let's look at, for example, a go, no-go task. So every time the animal breaks a beam here, now we have to differentiate between two different sounds, okay, or smells in this case, whatever. Every time the animal breaks in the beam, he can give one of two answers, but there are one of four outcomes. Okay, let's say in this task, I'm teaching the animal to discriminate between one stimulus to another stimulus. And this is the correct tone, and this is the wrong tone. We call it the non-target tone. And the animal must lick only in the target tone and avoid licking when it hears the target tone. Okay? So every time the animal breaks a beam, he hears one of two sounds. We now want to understand the perception of these sounds. We'll see it a little bit in the next slide. But let's look at the outcome. So if he hears the target tone, the response of the animal can either be to lick or not to lick, one of two options. But we have four options when we hear this and when we hear this. When we hear the target tone, if it licked, we call it a hit. In this case, the animal gets a water reward. If it did not lick, we call it a miss. It was supposed to lick, but it's a miss. If it heard the non-target tone and licked, this is the most horrible scenario. It will get a punishment, and we call it a false alarm. And if it heard the non-target tone and did not lick, it did the right thing, and we call it correct rejection. Great. Okay, so there are four outcomes to this type of learning, and these animals can very rapidly learn. This is just an example from work in our lab. Before they get a punishment, they lick in very high percentages, and then when they first get a punishment, this falls to 50% chance or lower, and the performance of the licking in the non-target tone is shown in red, and in blue is licking in the target tone. And you can see that mice very rapidly avoid licking the non-target tone and lick in the target tone. But there is some noise in the system. Okay? And in order to quantify that, we use what we call probabilistic statistics in the following way. We assume that the decisions, the distributions of the decisions is of the sensation that the animal feels have Gaussian probability, and we build this 2x2 matrix. And again, there is the stimulus that I give and the response that the animal responded. I give a total of 100 stimuli, which is target tone, and the total stimuli, which are non-target tone. Let's say it's red and blue. And you have to tell me, or the subject has to tell me, in this case it was the mouse. We can do, again, this thing in the mouse as well. He can tell me whether it was red or whether it was blue, or yes, no. It has two options, And generally, there are four possibilities. Okay, you can have a hit when it was red, and he told me it is red. We call it a hit. In this case, it was 65 times out of 100 that the animal got it right. He missed in 35 out of 100 times. When I showed him red and he said no, or he said it's blue, and he did it 80% correct when I showed him blue and he said it's blue, and only 20% false positives when I showed him blue and he said it's red. Okay, this must accumulate at 100 because I showed the stimulus 100 times, and of course the responses must add up to 200. Yeah. Let's say this is after he learned. Okay. Let's say this is after he learned. I want to now quantify the discriminability. I want to quantify the percept. And one way to quantify perception is to use probabilistic statistics. Okay. So what we assume, we can take this data and we plot it as Gaussian distributions on a plot like this, and the x-axis would be the sensation that is evoked by a given stimulus, and we divide it to the blue and the red events. Okay, in total I gave 200 stimuli, so each distribution would be 100 stimuli. Okay, and now we can we plot this way, and the overlap between the blue and the red here are the false positives. So this is what we call the decision boundary. I showed him red, but he said it's blue. So this is the false positive. This is the region here underneath the curve. The region shown in white is the misses. Okay, whenever he was supposed to say red, but he said blue. Okay, it's shown in white, and this is the correct rejections. So the correct answers that the animal gave is the hits and the correct rejections. And if we plot these two distributions as such, we can calculate a measure which is called d prime, which is basically the distance between the peaks of these two distributions. And this d prime is a quantitative measure of the discriminability of the animal. Exactly how well did the animal discriminate between a yes and a no. If these two distributions were far away with no overlap between them, then we would have very few false positives, very few misses. All the time it's easy. So for example, if I give you two stimuli, and they are very, very easy to discriminate between, then the d prime will be large. And the more that these distributions overlap with each other, the less the animal can discriminate them in a less efficient way. Okay, so this is something that we can quantify in a measure called d prime. Now the d prime value is not sensitive to biases. So for example, you can see that these two distributions are shifted to the left with regard to the decision boundary. And where they overlap is what we call the ideal observer. And if there's no biases in the task, it will fall directly on the decision boundary. But if we do have biases, like the ones I mentioned earlier, okay, if I have an incentive to say that something is correct, if it's fuzzy, it's around the decision boundary, then I will have more false positives or more false negatives depending on the bias of the behavior. Okay, so this is a very powerful way to quantify perceptions, which is something that we use to date in animal research. Okay, and clearly false positives increase when noise exceeds response thresholds. We make more errors. And like I said, this d prime is insensitive. Okay, it provides the separation between the means of the signal and the noise, and it can be thought of as the sensitivity of the detector. How well does the animal perform the task? And the higher D is better detection, and it's an accurate measure that compensates for the differences in the behavioral biases. Okay. Yeah. So basically, you would say, using the regular method that you suggest, you would say that this animal was correct 65% of the time. Okay? That would be your measure of percent correct. But what I'm saying is that there are biases in the system that shifts the decision boundary around, and using these measures, it's around the average between the 65% and the 80%. So it's around 73%. So if I ask you how well did this animal do, you can say either 65% or 80% or the average of both. But this way of plotting the normal distributions gives you a more accurate measure of the detectability. Okay, it's a measure called D prime, which is insensitive to the biases because it takes all four measures into account: the hits, the correct rejections, the misses, and the false positives. So in many cases in papers, you will see that people do plot the correct, the percentages of the correct, of the hits and the false positives or something like that. In fact, I just showed it to you in the previous slides. Okay? This 80% is how much he said hit out of the 100. So that would be 80. And this is how many mistakes he made, the false alarms that he made, which is very low. But in order to give you an accurate estimation, which is insensitive to the bias, D prime is a better measure. So for this animal, that would be a D prime quite high because it's doing quite well. Something on the order of 2.5 or 3 would be the D prime. And it's insensitive to these behavioral biases. Mm-hmm. No, so the concrete example here is that you would say that it's 65% correct. That's what you would normally do. Okay, let's calculate how much he's correct here. Okay, but in fact he is doing better than 65%. That's what I'm telling you. Okay, because he has biases that are built into the decisions that the person has to make. And the more accurate way to measure this, if I have two people who will have different numbers in here in terms of the percent correct, can have the same D prime values, and that means that they have exactly the same discriminability of this sense. Is the distance between the peak distributions of the average will be somewhere closer. So taking the average between these two is better than taking only one. But a single measure that takes into account, which is normally used, is this measure that is called D prime. And you can have people with different biases. Okay, so we'll have completely different measures. And this, we can give some concrete examples that you'll see that the numbers will change a little bit. But the average is better than taking only one. And the D prime is the cleanest way to get that. Because these can shift all around. If it will be on exactly on the boundary, then you can use the regular measures. Yes?
Also, the variance, like they could be thinner or thicker.
Mm-hmm.
So it's also, like if you use only the averages, you don't take into account the variance, the standard deviation.
How is the variance? How is the variance accounted here? What units are these?
So we assume that there is a Gaussian probability of the responses. That's one way to measure, which is common in the field.
Gaussian distribution with what variance? I'm saying you can have the same D prime value
Yes.
different with narrower Gaussian, it will create different confusion related to success.
Like this, yes. But you'll have the same D prime.
Exactly.
Mhm.
But these are not the same perceptual. I don't know.
No, it will have. It's exactly the same discriminability of the subject, even with different variances.
But it will have. It will. You have like four. You have four constraints here in the confusion rate, even three, and you have five parameters that you need to choose in this model. You have the mean of the two Gaussians. You have
Yeah.
the variance of the two Gaussians.
Yes.
You have the decision boundary.
Uh-huh.
So you can actually you have five parameters and only three constraints. You can create everything you want. I'm asking whether
Okay.
the variances should be the same or the variances should be one.
Well, in this case, the variances are the same in this example, but you can see different. You can see measurements with different variances.
How
But
can you
if
say
it's
that?
normal, it's distributed, so the D prime is not supposed to be affected by the variance because if it's symmetrical, so that doesn't matter.
Bon. Okay, so these are measures. These are ways to quantify perceptions that you will see in the literature. Okay. Another way to calculate this or variations of this is the ROC, which you also use a lot in receiver operator characteristic that you can also see in the literature when we go along. Okay. We're done. One last thing, one last word. Also, if you look at sensory systems, one thing which is very common is speed-accuracy trade-off. The accuracy is higher when you have more time to sample the sense, and this can be measured by the time that that the subject is, when you ask a subject to detect a stimulus, and if you give him more time to sample, you can see increased accuracy, and this is something that people use a lot, response times in quantifying perceptions, okay? If I ask you whether you heard the stimulus or not, and the response time of the subject is something that is used in many cases for quantification of the perception. Bon, so we meet again on Wednesday to continue with basics on sensory systems, and then if I have time, I give you some basics on how to give a lecture and what to avoid, and then we jump into the water with different teachers that give you different aspects of sensory systems. Bon, have a nice day.
כן, שיעור לא.
להבין בקורטקס איך הקורטקס מגיב לגירוי של התוכנית. מגלים שאזורים פה בקורטקס יש גם מגמת השכם הזה, יש פה גירוי של התוכנית. בואו נגדיל את זה. במוח רגיל של עכבר רגיל, במוח רגיל של עכבר רגיל, גירוי של שערה אחת גורם לאקטיבציה כזאת. תקחו את הכוח בה, שחררו אותו לחופשי. אוקיי? תפסו אותו אחרי חודש. תגלו שאותו גירוי גורם להפתרה מאוד מאוד משמעותית בשדה הרפלקטיבי של התאים.
זה הסתרה או שזה בתיאור תגובה?
זה מה שאת אומרת.
זה הרבה יותר גירויים בכל התהליך.
לא יודע, אני אומר לך, הנה דוגמה למה שאת אומרת, שאכן קורה במציאות. לקחת חיות שהן ניסיון חיים, חוות הרבה חוויות, המוח נראה שונה
בעקבות זה מה יהיה פה?
לא, יש אנשים כמוני שרוצים את העכברים. עכשיו מנהיגים אותי למשהו אחר. כל מה שרוצים לעשות מהעכברים שלך מאיתם. זה העכבר שלך, העכבר השני אותו דבר, העכבר השלישי אותו דבר, עם איזשהו וריאנט ביולוגי ביניהם, מעט אבל. ואז אני מלמד את כולם, חצי מהקבוצה אני מלמד לעצמך, חצי מהשני, ואז אני יכול להגיד משהו אקוויוולנטי על השינויים שאני רואה. אם הייתי לוקח את החבר'ה, אם כל עכבר יש לו